Ever wondered how the future of DNA cloning might shape our world? Imagine a universe where we can replicate life forms, where we can control the blueprint of life itself. DNA cloning, a concept that once belonged to the realm of science fiction, is now a scientific reality. Yet it's a world filled with both promise and paradox. The ethical implications and scientific considerations are as fascinating as they are complex. Join us as we journey through time, exploring the captivating advancements in DNA cloning. Our journey begins in the mid-20th century, where the foundations of cloning were laid. During this pivotal era, two brilliant minds, James Watson and Francis Crick, unveiled the double helix structure of DNA, the very blueprint of life. This groundbreaking discovery marked a turning point, opening doors to the fascinating world of genetic manipulation. The first attempts at cloning were met with a myriad of challenges. Scientists grappled with the intricate task of replicating DNA, a process that was as delicate as it was complex. Despite the hurdles, their relentless determination propelled the field forward. They toiled in their labs, their eyes set on the horizon of possibilities that cloning offered. These initial advancements, while humble in their beginnings, were nothing short of monumental. They sparked a new era of exploration, setting the scientific community ablaze with curiosity and ambition. These early steps set the stage for a scientific revolution in the years to come. Fast forward to the 70s and 80s, a period of groundbreaking discoveries and advancements. This was an era when the world of DNA cloning really began to take shape. It was during this time that scientists made one of the most significant breakthroughs in the field, the successful cloning of a gene. In 1973, Herbert Boyer and Stanley Cohen achieved a feat that changed the face of genetic engineering forever. They took a piece of DNA from one organism and inserted it into the DNA of another, effectively cloning the first gene. This was a monumental step that moved us closer to understanding the blueprint of life. It was an achievement that paved the way for future advancements in cloning and genetic engineering. But the 70s and 80s were not just about gene cloning. The period also saw the birth of one of the most famous animals in the world, Dolly the sheep. Dolly was not just any sheep. She was the first mammal to be cloned from an adult cell. This was a breakthrough of epic proportions, as it challenged the scientific belief that adult cells could not be used to create an entirely new organism. The creation of Dolly in 1996 by Ian Wilmut and his team at the Roslin Institute was more than just a scientific curiosity. It was a revelation that pushed the boundaries of what was possible in the world of cloning. The implications were enormous, opening up new fields of research and raising complex ethical and societal questions. These breakthroughs not only challenged our understanding of genetics, but also opened a Pandora's box of possibilities. In the span of just 2 decades, we moved from cloning simple genes to cloning entire organisms. This was a time of rapid progress and unprecedented discoveries, a period that truly laid the foundation for the world of DNA cloning as we know it today. The new millennium brought with it a wave of innovation and progress in DNA cloning. One of the most groundbreaking advancements of the 21st century has been the development of therapeutic cloning. This procedure aims to create embryonic stem cells, which can be used to replace or repair damaged tissues and organs. Imagine the possibilities if we could grow our own replacement organs. No more waiting lists for transplants, no more organ rejection. Another significant stride in this century is the creation of synthetic life. Scientists have successfully synthesized the genome of a bacterium, effectively creating a new life form from scratch. This isn't just about playing God, it's about understanding the very essence of life itself. With this knowledge, we could potentially design organisms tailored to perform specific tasks, like cleaning up oil spills or producing biofuels. But the real game changer of the 21st century has undoubtedly been CRISPR technology. Short for Clustered Regularly Interspaced Short Palindromic Repeats, CRISPR allows scientists to edit genes with unprecedented precision. Think about it as a pair of molecular scissors that can cut and paste DNA sequences. This technology could potentially eradicate genetic diseases or even enhance human capabilities. The applications of these advancements in DNA cloning are not just limited to human health. They also hold great promise for agriculture and livestock breeding. For instance, Cloning could help us develop crops that are more resistant to pests and diseases, or livestock with desirable traits. This could revolutionize our food systems and contribute to global food security. But with great power comes great responsibility. The ethical implications of DNA cloning are complex and far-reaching. As we continue to push the boundaries of science, we must ensure that we do so with caution and respect for the sanctity of life. These advancements have brought us closer than ever to unlocking the full potential of DNA cloning. As we look to the future, one might wonder, what's next for DNA cloning? The answer to that question is as exciting as it is complex. Today, we stand on the precipice of a new era in genetic research and biotechnology. Advancements in DNA sequencing and editing technologies like CRISPR are opening doors to possibilities that were once the stuff of science fiction. Imagine a world where we could clone extinct species back into existence, or create personalized medicine tailored to an individual's genetic makeup. The potential applications of DNA cloning are vast and varied, from agriculture and conservation to medicine and bioengineering. But alongside these exciting possibilities come important ethical considerations. How will we regulate the use of these technologies? What are the potential risks, and how can we mitigate them? In the medical field, for instance, the prospect of cloning human organs for transplantation is both thrilling and fraught. It could save countless lives, but it also raises questions about consent, ownership, and potential misuse. And in the realm of conservation, while resurrecting extinct species could help restore ecosystems, it could also disrupt existing ones. While we can't predict exactly what the future holds, we can certainly expect DNA cloning to be at the forefront of many scientific breakthroughs. We must navigate this complex landscape with care, balancing the potential benefits with the ethical challenges. While the future of DNA cloning holds immense potential, it also poses intriguing questions that will continue to engage scientists and society alike.